they are saying an example of differential and selective medium in which colonies of gram-negative bacteria produce large, so gram-negative bacteria, they produce large amounts of acidic products and appear green with a metallic sheen is. So first we have to know what is differential medium and what is a selective medium. So differential media refer to the type of growth media that allows differentiation of closely related microorganisms. So it will allow the growth of some kind of microorganism uh, and it will not allow the growth of other. So they are also known as indicator media, and the differentiation media or differential media, depends, it actually is relies on the principle or it utilizes some biochemical characteristics of an organism, uh, which can be differentiated and it can be used as an identification of that particular microorganism from a group of other microorganisms. Okay, like you have a lactose fermenter and you want to isolate lactose fermenters particularly from a soil samples. So in soil there would be many different kinds of bacteria and you want to particularly isolate some lactose fermenters. So you can use differential media which will allow the growth of lactose fermenters, or you can use that media to differentiate between lactose fermenters and non-lactose fermenters. And then we have selective media, which refers to a type of growth media that allows growth of selected microorganism in that media. For example, if particular microorganism is resistant to particular antibiotic like tetracycline or ampicillin, how that can be possible if we have inserted or transformed that microorganism with a plasmid containing tetracycline or ampicillin, and then we plate that bacteria on a agar which contains that particular antibiotic. So it will only allow the growth of those bacteria which contain the resistant plasmid, and it will not allow other bacteria which do not have the plasmid, they will be killed. That antibiotic can be added to the medium, prohibiting the growth of other bacteria in that medium. So this is the difference between differential and selective media. So both these types of media are used to select for our desired microorganism from a group or a sample which contains other microorganism as well. So here the question says an example of differential and selective media in which colonies of gram-negative bacteria. So only gram-negative bacteria will grow in that, and then these bacteria also produce large amounts of acidic products and appear green with a metallic sheen. So which of these agar or which of these medium does that? So we'll go through each of these in detail. So first is the blood agar. So blood agar, and okay, before that we'll also the different types of differential media. First is blood agar, and what is its significance? It contains bovine heart blood that becomes transparent in the presence of hemolytic substances which are released by the bacteria. Next is the streptococcus ethylene blue or EMB. It has the significance is that differentiation of lactose and sucrose fermentation. So the bacteria which can ferment lactose or sucrose can grow in this. Then we have McConkey. It is differentiation for lactose fermentation, and mannitol salt agar, differentiation for mannitol fermentation. Another is the X-gal plate, which is used for cloning and differentiation of blue-white colonies for Lacoperon mutants. And we also have selective media, which consists of eosinmethylene blue for gram-negative bacteria, YN for yeast and mold, McConkey agar again. It allows the growth of only gram-negative bacteria, not the gram-positive. Even this HE medium also gram-negative bacteria. Mannitol salt agar, on the other hand, is used for selection of gram-positive bacteria. Terrific broth for recombinant strains of E. coli, and XLD for gram-negative bacteria. So in the question, first of all, they have asked for gram-negative bacteria, and here we see that mannitol salt agar, it is used for gram-positive bacteria. So straight away, this mannitol salt agar, we will straight away reject this option, and the other three we can look upon that. Here, EMB and McConkey, we see that they are important for gram-negative bacteria. Here we can see EMB and McConkey, they both are for gram-negative bacteria. So it either of the two can be our uh, desired media. So within that also, blood agar is also there. As I mentioned earlier, that blood agar is used. It contains the medium contains blood, so and it can differentiate the bacteria which releases certain toxins or byproducts which are capable of destroying the red blood cells. Blood agar is a general purpose enriched medium which used to grow fastidious organisms and to differentiate them based on their hemolytic properties. So the bacteria, these bacteria, they release hemolytic substances, and so the RBC is lysed in those area where the bacteria is present, and they appear whitish. Okay. Next is the EMB agar. So here you can see in the picture only you can see there is a metallic sheen, green metallic sheen is there. And I have already mentioned that it is a selective and differential media used for isolation of gram-negative bacteria. So another criteria is also fulfilled, that gram-negative bacteria only grows here, and it also shows the green metallic sheen. So as a selective media, another points to take note is that it is contains inhibitors such as eosin and methylene blue that suppress the growth of gram-positive bacteria. Therefore, only gram-negative bacteria will grow. And as a differential media, it can differentiate between different types of gram-negative bacteria on their ability to ferment lactose. So, what are this differentiation? So, so the strong lactose fermenters, like E. coli, it is a strong lactose fermenter and it produces colony with green metallic sheen. Here you can see this is a green metallic sheen. It is a strong lactose fermenter, so it will ferment high amount of lactose. Then we have weak lactose fermenters that are slow fermenting lactose and they produce brown or pink colonies. Here they are brown or pink. And then we have the non lactose fermenters such as Shigella, Salmonella, and Enterobacter, as it is shown here. They do not produce acid and they form a clear, colorless, or amber colonies that can be distinguished from other ones. So, say suppose you take a sample from a water body or a soil and you streak that sample on this ENB agar, and then you can differentiate between different kinds of bacteria based on their color that appears on that ENB agar. So, and lastly, the other two types of agar which the question talks about is the McConkey agar. So this is also used to grow gram-negative bacteria, but it appears like this, and there is no green metallic sheen in this McConkey agar. The lactose fermenters turn red or pink in McConkey agar, and not non-fermenters do not change the color. So these are the non-lactose fermenting; they will appear colorless, and lactose fermenting will appear pink or red. Lastly, there is mannitol salt agar, which is for the isolation of gram-positive bacteria like Staphylococcus. So, Staphylococcus here, Staphylococcus epididymis, it is a non-pathogenic strain that is normally present on our skin also. Epididymis, that is present on the epidermis layer of our skin. And another is Staphylococcus aureus, that is the pathogenic strain. So this pathogenic strain will turn the mannitol salt agar colorless, and that is how we can isolate it. And both these are gram-positive bacteria. So it will support the growth of gram-positive bacteria only, not the gram-negative bacteria. So based on all this discussion, so we have the medium which in which colonies of gram-negative bacteria. So gram-negative bacteria is in ENB and McConkey, and which produce large amounts of acidic product and appear green is EMB. Not McConkey because it will turn red or colorless. Not mannitol because it supports only gram-positive bacteria. And blood agar is the colonies appear white if they can lyse red blood cells. So the correct option is option B, EMD agar or eosin methylene glucose.